for today's presentation, there will be three main parts. First, I will explain the physiology and pathology of the pancreas, followed by a description of the PI3K signaling pathway. I will then state the objectives of the PhD. The experimental results we presented in two parts. The first part will focus on the role of EPS34 in acinar cell physiology, and the second one on the role of PI3K alpha in pancreatic cancer progression. Finally, I will present the conclusions of the project. So the pancreas is an organ that is located in the abdominal cavity. It sits behind the stomach. It is surrounded by different organs such as the liver, the small intestine, and the spleen. The thicker part of the pancreas is known as the head, and it sits at the juncture where the stomach meets the small intestine. The middle part is known as the body, and the thin end is known as the tail. The pancreas belongs to the digestive and to the endocrine system. It has an endocrine and exocrine functions, and it plays a key role in digestion and glucose homeostasis. The endocrine pancreas comprises 5% of pancreatic mass. It is composed of islets of Langerhans, which are islets composed by different cell types which produce different hormones involved in glucose homeostasis. The exocrine pancreas comprises 95% of pancreatic mass. It is composed of acinar cells, which are highly polarized cells that possess different functions at the apical and basolateral domains. These cells are in charge of secreting proteases in zymogen, such as trypsin and chymotrypsin, as well as digestive enzymes such as amylase and lipase. Other cells that form part of the exocrine pancreas include duct cells, central acinar cells, and pancreatic stellate cells. So because of the nature of the pancreas, there are different pathologies that can originate at any of its compartments. From the exocrine pancreas, we have pancreatitis and pancreatic ductal adenocarcinoma. For the endocrine pancreas, we have diabetes and neuroendocrine tumors. In this presentation, I will only focus on the diseases of the exocrine pancreas. And I want to mention that from now on, whenever I mention pancreatic cancer, I will be referring to pancreatic ductal adenocarcinoma and not to neuroendocrine tumors. So pancreatitis is the inflammation of the pancreas. It can be in two different forms, as acute attacks, but characterized by sudden inflammation and in most of the cases, kind of reversible damage. If the inflammation does not resolve, the condition progresses to a chronic state where there is irreversible damage to acinar cells and to intrapancreatic nerves. Having chronic pancreatitis increases the risk for developing pancreatic cancer. So there are many risk factors for developing pancreatitis. We still don't know its right origin, but there are several cellular processes that could be at its cause, such as zymogen activation, mitochondrial dysfunction, endoplasmic reticulum stress, and defective autophagy. The studies that have used knockout for different proteins involved in the autophagy pathway have resulted in chronic pancreatitis, highlighting the importance of this pathway in maintaining the acinar cell functions. In case that acinar cells undergo any of these problems with these functions, there is an activation of the inflammatory cells. As I mentioned, if this inflammation is not resolved, we would end up with chronic pancreatitis. So pancreatic ductal adenocarcinoma represents 95% of all cases of pancreatic cancer. It is a highly lethal cancer. It occupies the 4 leading causes of cancer-related mortality and is thought to become the second world leading cause by the next decade. It has a really poor prognosis with a 5-year survival of less than 5 years. Most of the time, it is detected at late stages. Its current treatments include surgery, but it is only applicable to 15 to 20% of the patients. Chemotherapy, it remains the only option for the rest of the patients. Pancreatic cancer is a really heterogeneous cancer. There is a dominating mutation, which is the activating mutation of KRAS, which is present in around 95% of all the cases, followed by the loss of suppressor tumor genes such as TP53, CDKN2A, and SMAD4. Because of this heterogeneity, there have been efforts to try to classify the different types of pancreatic cancer, but the current histopathological classification remains largely uninformative regarding the therapy management for this cancer. It is mostly used for diagnostic purposes for determining the stage of the cancer. So next generation sequencing and different scale technologies have allowed us to molecularly subtype pancreatic cancer. By molecularly subtyping pancreatic cancer according to its genetic alterations, we could have better tools for selecting the patients, for selecting the better treatment for them. This would also provide clinical relevance on how to treat these patients. So molecular subtyping is still at its early steps, and it's not currently used in clinical practices. So the development of pancreatic cancer, there is still a debate on which cell is at its origin, but many studies suggest that acinar cells are the culprit. So under inflammatory signals, stress, and the activation of KRAS mutation, normal acinar cells undergo the transdifferentiation into acinar to ductal metaplasia. In case that the inflammatory signal is continued, this process becomes irreversible, and the cells progress to low-grade pancreatic lesions. In addition to the inflammatory signal, the loss of tumor suppressor genes further exacerbates this progression towards a higher degree of lesions until we end up having a carcinoma in the pancreas. As I mentioned before, KRAS is a major event in pancreatic cancer. Oncogenic KRAS controls two main affects two different pathways, the MAP kinase pathway and the PI3K-AKT pathway. The PI3K-AKT pathway is overactivated in at least 50% of cancer patients, and particularly in pancreatic cancer, it denotes a really poor prognosis. Before I ponder on the possible targeting of PI3K in pancreatic cancer, I will explain what PI3Ks are. So, phosphatidyliniositol, also known as PI, is a lipid composed of a glycerol backbone, an inositol ring, and two acyl chains. The inositol ring can be phosphorylated at 3 different positions, 3, 4, and 5. The different phosphorylation of this lipid can give rise to 7 different lipids, which all play different roles in cell signaling. For this presentation, I would only focus on the enzymes that phosphorylate these lipids at the 3 position, the PI3 kinases. So PI3 kinases are grouped into 3 different classes, class 1, class 2, and class 3. For class 1, we have 4 different heteromeric enzymes, which are composed of a P110 catalytic subunit and a P85 or P101 regulatory subunit. The PI3K alpha and beta are expressed ubiquitously, and PI3K delta and gamma have a restricted expression to the immune system. They phosphorylate PIP2 into PIP3, and they have been involved in different cellular processes such as translation, cell growth, glucose metabolism, cytoskeletal motility, and cell survival. For the class 2, we have 3 monomeric enzymes, C2 alpha, C2 beta, and C2 gamma. The C2 alpha and C2 beta are ubiquitously expressed, and C2 gamma has a restricted expression to liver, prostate, and breast. They can phosphorylate PI into PIP3, but also PI4P into PIP2, sorry. Their role are still unknown, but they have been implicated in growth, cell migration, glucose metabolism, angiogenesis, and cell survival. For class 3, we have only 1 member, which is known also as BPS34, and it forms an heterodimer with BPS15. It is mostly found in protein complexes such as complex 1, which regulates autophagy, or complex 2, regulating endocytic sorting. This lipid is the BPS34 phosphorylates PI into PIP3. So class 1 PI3Ks can be activated by receptor tyrosine kinases, GPCRs, or RAS. After activation, they phosphorylate the lipid into PIP3, which then is going to interact with other effector proteins and induce different signaling cascades. These lipids are mostly produced at the plasma membrane. For class 2, the mode of activation is still unknown, but what we know is that these lipids are mostly produced at the intracellular level, where they regulate different endocytosis and trafficking pathway processes such as clathrin-mediated endocytosis. Besides their catalytic function, they also have scaffolding functions, such as the case of C2 alpha, which participates by stabilizing the mitotic spindle. The action of BPS34 is needed at the initial stages of autophagy and for the production of the endosomes and phagosomes. BPS34 is also needed to produce PIP3 pools at the later stages of these two processes to promote the fusion with the lysosome. Besides this function, BPS34 also has a scaffolding function by stabilizing the other proteins from these two complexes. So the oncogenic roles for class 1 and class 1 PI3Ks are the most implicated in cancer. For class 2 and class 3, we still don't know if they could have an oncogenic role. So in class 3, there are activating mutations in PIK3CA, which is the gene that codes for the P110 alpha catalytic subunit. These mutations are mostly found at the helical domain and at the kinase domain. It is highly mutated in different solid cancer, but in pancreatic cancer, it is present in less than 5% of the cases. For the other different isoforms, there have been amplifications and overexpression in certain cancers. So as we know, the PI3K-AKT-mTOR pathway is highly targeted in cancer, and the first inhibitor for the PI3K isoform that was developed and accepted for use was the selective inhibitor for PI3K delta. Recently, an inhibitor for targeting PI3K alpha has been approved for treating metastatic breast cancer. This inhibitor is known as BL719 or alpelisib, and it's given to patients that present PIK3CA mutation, HER2 negative, and HR2 positive metastatic cancer, breast cancer. For pancreatic cancer, there are two clinical trials of phase 1 have just been recently finished. The official results are not yet published, but in this case, there was no patient strategy. So considering what I have told you, that the PI3K pathway is overactivated in pancreatic cancer, we set the objectives of my PhD. So basically focusing on the class 1 and class 3. So one of the objectives was to determine the role of BPS34 in acinar cell physiology, and the second one to characterize the role of PI3K alpha in pancreatic cancer progression. So for the role of BPS34 in acinar cell physiology, I generated a mouse model where the BPS34 catalytic site had been deleted by removing the exon 21. So from the conclusions from this part, we have that inactivating BPS34 in acinar cells disturbs their cell homeostasis. It seems to induce pancreatic steatosis. We still don't know if it's a fatty replacement of the pancreas or a transdifferentiation of acinar cells due to this effect. But not less, we observe that this induces a chronic pancreatitis phenotype in these mice. Now I will go on describing the results of PI3K alpha in pancreatic cancer progression. So this study was based on previous results from the lab that had demonstrated that the PI3K alpha isoform was responsible and crucial for initiating pancreatic carcinogenesis. Now, we wanted to know if this same isoform could have a role in pancreatic cancer progression. So the first part of the study consisted on analyzing patient databases where we analyzed the expression of 50 hallmarks that could be altering normal chronic pancreatitis, localized or metastatic pancreatic cancer patients. What we observed was that in pancreatic cancer patients, there was a higher expression and activation of the PI3K-AKT signaling. So, further analyzing the PI3K-related reaction between localized and metastatic patients, we observed that there's a higher activation of the PI3K cascade towards FGFR2 in metastatic patients, which would suggest that in this patient there's a differential activation of receptor tyrosine kinases. So we know that PI3K alpha is the main PI3K isoform activated downstream of receptor tyrosine kinases. So taking that into consideration, we created a PI3K regulated signature of different genes, and all regulated by PI3K alpha, and this signature allow us to clusterize normal chronic pancreatitis, localized and metastatic cancer patients. We also observed that metastatic patients presented a higher activation of this subset of genes. We then use this signature and apply it into two different patient cohorts. And what we observed that a higher activation of the signature in any of these patients regarding their pancreatic cancer stage denoted a worse overall survival and prognosis for these patients. When we analyze only localized patients that had been deemed as localized, we observed that the signature was able to predict that those that were prone to relapse or could have a worse survival. So we then try to do an in vitro approach. So for this, we use a panel of PI3K inhibitors that were isoform selective or pan-PI3K inhibitors. We tested them on a panel of cell lines from murine and human origin that had a different mutational status for KRAS, P53, P16, PIK3CA, and P10. So we tested them and we observed their effect on different parameters such as AKT phosphorylation, where we observed that only PI3K alpha selective and certain pan-PI3K inhibitors had the same effect on the phosphorylation of AKT at the serine of 473. We also observed their effect on actin remodeling, cell motility, cell migration, and cytotoxicity. In all the cases, what we observed is that only PI3K alpha selective inhibitors and certain pan-PI3K inhibitors can have an effect on any of these parameters. So we then use a genetic approach where we use a scrambled siRNA targeting PI3K alpha, siRNA targeting all the PI3K class 1 isoforms, trying to mimic a pan-PI3K inhibitor. And what we observe is a similar effect on migration. When we use, for example, shRNA, what we observe is that only targeting PI3K alpha and not PI3K beta has an effect on migration. This validates our previous pharmacological findings where PI3K alpha seems to be the isoform that drives migration in pancreatic cancer. So trying to explain a bit more why we were having this effect with pan-PI3K and the alpha selective inhibitor, we looked for a concentration of the inhibitors where they both had the same effect on the phosphorylation of AKT. And at 1 micromolar, we do not observe a difference in the percentage of living cells when treating them with either inhibitor. However, on migration, we observe a drastic and significant decrease on the migration of cells that were treated with the PI3K alpha inhibitor. So one possible explanation could be that these inhibitors were maybe perhaps changing the distribution of the different PIP3 subspecies. So by measuring the lipid subspecies, we observed that the general PIP3 subspecies, we observed that only PI3K alpha can decrease the percentage of total PIP3s. When I mention PIP3s, different subspecies, what I'm mentioning is that, okay, so these lipids are all PIP3s, but the subspecies correspond to the different acylation of these chains. So what we observed was that PI3K alpha selective and pan-PI3K inhibitors induce a different distribution of the different subspecies of PIP3. This difference of the distribution could also explain why they were having a different effect on this on migration. So we then wanted to know if the genetic background of the cell lines could also be influencing the response to the inhibitors. So for this, we calculated the correlation between the in vitro IC50 of the different inhibitors and their capacity to inhibit migration. What we observed was that the more an inhibitor could target PI3K alpha, the more effect it had on migration. The same was not observed for any of the other isoforms. So therefore, a PI3K inhibitor must strongly target PI3K alpha to have an effect on cell migration. So here we plotted the p-value of the correlation of the IC50 for each different isoform. And what we can observe is that regardless of the different mutational status of the cancer cells, or the pancreatic cancer cells, it is the PI3K alpha isoform which drives migration in all the cases. And here we can observe that for these cell lines, all the PI3K delta might also play a role, but alpha is still driving, also has an effect on migration. We wanted to test Also, we tested the same thing for some the cytotoxicity. Sorry. So we tested the same thing using different cell lines from different organs, and what we observed was not the same. So what we observed was that for other organs, that means also for other cancer, the different PI3K isoform that it's going to drive migration will change depending on the organ and on the mutational context. So we then measure the PI3K activity in tumors from pancreatic cancer patients. So by using phospho-AKT substrate staining, we were able to detect higher PI3K activity in tumors that from patients that also presented higher levels of cell-free DNA and consequently of circulating tumor DNA. We thought that perhaps there might be a correlation between the PI3K activity and the high cell level of the circulating DNA. So, as you know, cell-free DNA, it comes from the nucleosomal fragmentation due to inflammation, necrosis, or apoptosis. There is a small fraction of cell-free DNA known as circulating tumor DNA, which is thought to come from cancer cells. In pancreatic cancer patients, detectable levels of circulating tumor DNA are correlated to a worse prognosis and are considered sometimes as a sign of micrometastasis. So, for our in vivo model, we use the KPC mouse model is a mice that presents the oncogenic mutation of oncogenic activating mutation of KRAS and the loss of function of the TP53 expressed in pancreatic epithelial cells. So the prognostic value or any value for cell-free DNA in KPC mice has not been reported. So by measuring the cell-free DNA by using two different genes, we measure its relative concentration, and what we observed was that KPC mice that presented higher levels of cell-free DNA also had shorter lifespans in comparison to mice presenting lower levels of cell-free DNA. So that we could say that in circulating cell-free DNA, KPC mice has a prognostic value. So trying to correlate the levels of cell-free DNA with disease progression, what we observed was that it matched the progression of the disease. Mice having lower levels of cell-free DNA, in some cases, only had like the tumors, in situ tumors, but no micrometastasis as mice presenting higher levels of cell-free DNA. We also, I tried to characterize the integrity of the cell-free DNA that we were detecting. And for this, we found that there was a fragment that was between 160 and 210 base pairs. This fragment also recapitulated our previous finding with circulating cell-free DNA. So from this part, we have that cell-free DNA in KPC mice correlates with disease progression and has a prognostic value. We then set a diagnostic protocol where we use ultrasound imaging and the analysis of cell-free DNA. Once that we had detected a tumor and an increase in the levels of cell-free DNA, we gave the mice a treatment using a PI3K alpha inhibitor, BIL-719, for 2 weeks. So what we observed was that in tissues from mice treated with the alpha-specific inhibitor, we observed a decrease in the phosphorylation of AKT in all the tissues and only a decrease of ERK in the pancreas. We also observed that mice that were treated with BIL-719 had a slower tumor progression. They also had less micrometastatic foci and lesser metastatic areas, which could which could be summarized as a reduced tumor evolution and metastatic progression. So these mice that were treated with the PI3K alpha selective inhibitor also had a delayed development of ascites. In fact, only just one mouse presented ascites in comparison to 4 from the control group. These mice also had longer survival than the other mice. So, besides these effects on delay in the tumor evolution, we also observed a decrease in the proliferation of cancer cells at primary tumor and at metastatic sites. Trying to validate the effect of this drug on metastasis, we performed a tail vein injection where we injected pancreatic cancer cell lines that had been engineered to express luciferase at the tail of the nude mice. and we then treated them for 3 weeks with BIL 719. What we observed was that the mice that were given the treatment had less cell proliferation, as denoted by the photon count, in the lungs. We could then say that treating the animals with this drug prevents the metastatic development. So we also know that in pancreatic cancer, macrophages play a really important role. So they are involved in tumor progression, and they also have a role in metastasis formation. We then wanted to analyze the effect of inhibiting PI3K alpha on this cell population. So by analyzing the pancreatic tissue, peritumoral tissues, what we observed that there is not a difference in the count of the F480 macrophages, but there is a decrease in CD206 macrophages, which could be summarized as that BIL prevents the pro-tumor macrophage differentiation. So we also tried to observe if oncogenic PI3K alpha could mimic the effect of oncogenic KRAS on macrophages. So we used 2 different mouse models. That this one, for example, I was expressing KRAS in acinar cells, and this one had the oncogenic activating mutation of PI3K alpha in acinar cells. We do not observe differences in terms of general macrophages or in CD206 macrophages. Therefore, oncogenic PI3K same effect as mutant KRAS on macrophage differentiation. So we then wanted to validate the pharmacological inactivation of the PI3K alpha with its genetic inactivation and their effect on macrophages. So we used another mouse model. This time was a KPC mouse model, but with inactive B110 alpha catalytic subunit. It's important to mention that these mice do not develop high-grade lesions or tumors. So when observing low-grade lesions, we do not observe a difference in the number of CD206 macrophages. However, when we quantify these macrophages around high-grade lesions, we observe that inhibiting by half, that means in the heterozygous mice, there was a decrease in the number of CD206 macrophages. So to summarize this project, we have that the PI3K alpha signature activation allows us to identify patients with under-detected micrometastatic disease. It also prevents the development of micrometastases into macrometastases, and it seems that PI3K alpha has an immunomodulatory effect. For the perspectives of this study, we'll include analyzing patient samples for an activation of the PI3K signature or for high levels of circulating tumor DNA before deciding to treat them or not with PI3K specific inhibitors. So to conclude this project, I have presented you 2 different models where I work with 2 different isoforms, PI3K isoforms. So for the PI3K isoform, we know that targeting PI3K AKT pathway will benefit the management of cancer. We also know that the PI3K inhibitors had an early failure in clinic because of their high toxicity, but this problem was partially solved by the development of PI3K specific inhibitors, but they still had a modest effect. Now we know that to have more benefits, we need to stratify patients according to the mutations and specific and other biomarkers. The challenge remains in detecting better selecting these biomarkers. And finally, from my work, and we can say that in pancreatic cancer, the use of PI3K inhibitions would delay metastasis. For BPS34, we know that it maintains acinar cell homeostasis, and that because of the heterogeneity of acinar cells, we end up with this mixed phenotype of pancreatic acidosis and chronic pancreatitis. But we still know if it has a role in preventing or promoting pancreatic cancer. So finally, to finish the presentation, I would like to say that when I started my PhD in 2016, I just thought that PI3K signaling was quite simple. It just consisted on activating a receptor and activating the different PI3K isoforms that would then produce the lipid, and the lipid will interact with different effector proteins, and that was it. So now I know that it's a bit more complex. So I know that I need to consider the sort of the signal rate, the duration, the strength, where it is located, the signal, in addition to considering the cell type, the developmental stage, the environmental, and the mutational status. And that it's not just the effector protein that is going to shape the signal, but also maybe different acylation here that it's going to affect the overall signaling processes. So I would like to thank all the members from my team and the people from the PI3K signaling pathway, and thank you all for your attention.
You